We were talking about conjugation when we left off, that's the third process. Remember, we have transformation, we have transduction, and then conjugation. One other little thought about transformation. Remember, that's the one that the DNA is just in the environment around the bacterium. It's not exactly known how that goes in, but it's thought that the bacterium um, that the DNA is going into has something in its DNA that recognizes it and pulls it in. But again, as I said, it's not totally known how, in terms of transformation, that bacteria is allowed to go into the new bacterial cell. So conjugation is, we're on the right group of pictures. There's four, looks like little rod bacteria. What you have is, what you're looking at in that circular purple, that's the plasmid. Remember, in bacteria you have a nucleoid region and then you have these plasmid rings. On these plasmid rings, there will be an area that codes for apilus and codes for the fact that this is the genetic male, as opposed to if you don't have if the other bacteria doesn't have this portion on their plasmid, they're genetically female. So we call this an F plasmid, F for fertility, because the F plasmid fertility is on the genetic male that we have described in past slides. And what happens is part of the plasmid gets nicked. And this is part of the process of conjugation. And then the portion of the DNA that gives it its F ability codes for a sex pilus. That sex pilus forms a bridge between the two bacterium cells, and that DNA unwinds. And so a strand of it, DNA is a double helix. So one strand will go into the new cell, and then of course the original donor cell can replicate its strand. So the strand that went into the new cell, and then actually there'll be a parting. So you do have eventually have the parting, but what will happen then is we're at the bottom in what's called C. The recipient, so what would be the female, is starts replicating on the transferred DNA, and that at replication then can move back into the host, or into the male cell. And so you do have at the end some transference of DNA between two different bacterium. The way we are looking at it color-wise, it might be helpful. You start out with just a purple plasmid ring. A portion of the purple goes into the recipient cell, and then that recipient cell starts creating a complement to that strand. That complement then goes into the original, what was the male cell, and now you have two cells, each with new DNA. So each with new makeup with regard to their DNA. And this is another way that DNA is transferred, through conjugation, a process where genetic material is transferred between bacterial cells. It incorporates the use of this fertility bridge or sex pilus, allows for the two cells to be pulled close together and actually join with regard to their plasma membranes, because this DNA needs to go past the plasma membrane, and there to be transference of DNA. So three ways that DNA can transfer from one bacterial cells to another. There's a fourth concept, and that is R plasmid. So I got a little bit ahead of myself. I forgot I had this particular slide. So this is just kind of reviewing what we just talked about, this F factor. The F factor is for fertility. um, is required. It gives that particular donor cell the ability to form the pili and donate the DNA. It exists as a separate plasmid, so it could be a plasmid F factor, or it actually can be incorporated into the chromosome. So it could be chromosomal F factor. So you actually have two different ways that this can happen. It can happen if a plasmid causes it to happen, or if there's a portion of the DNA itself in the bacterium that has this ability to make the sex pilus and then have the fertility for DNA to pass on from from the donor cell to the recipient cell. We tend to call the donor cell an F plus cell because the factor is present, a recipient cell an F negative cell because the factor is not present. The DNA is transferred during conjugation, and if it's transferred during conjugation, that then makes the recipient cell a donor cell. So it transfers the ability to be an F plasmid or an F chromosome is going to be transferred. This is one way that a bacterium becomes a recumbent bacteria. And you probably have heard this in news data, recumbent bacterias, so it causes diversity, causes again for there to be changes and adaptation with regard to bacteria. But I think we've gone through this. Step one is the pilus is created and attaches. Step two, one strand of the DNA transfers to the donor, to the recipient cell, from donor to recipient. Donor then synthesizes complementary strand, and the recipient then the recipient synthesized complementary strand becomes a donor cell itself. So you have this recipient cell going from a recipient cell to a donor cell at the end. The idea behind the R plasmids, I just want to say that that is for bacterial resistance, and if in that same plasmid ring, if there's a portion that codes for either slowing down how an antibiotic would work, or maybe it codes for putting markers on its surface that makes it, basically cloaks it, makes it invisible to bacterium, or it could actually cloak it to make the bacterium less effective, not work as fast, not walk as fast. So we call those R plasmids. So you have F plasmids for reproduction, R plasmids for resistance to antibiotics. Reproduction is one of our properties. The ability to take in energy and utilize energy to perform metabolic behaviors is another one. And there are various modes that prokaryotic cells use to obtain energy. One can be light, one can be chemicals, and some of them require very little, just carbon dioxide as a carbon source and minerals and maybe a little water. So the mode of nutritional uses separates one bacterium from another. The autotrophs are, you can have a photoautotroph or a chemical autotroph, and so these are prokaryotes that can create their own energy. They can make their own energy needs from a source that's outside of them. Photoautotrophs from light, their carbon source, the energy source is light, the carbon source is carbon dioxide. We think of them as the bacteria that can photosynthesize. We think of certain plants, for example, protists like algae. These are organisms, which algae we'll talk about next chapter, that have the ability to capture light as an energy source and carbon dioxide. Chemoautotrophs use inorganic chemicals such as sulfur and even nitrogen, plus carbon dioxide as the carbon source for their mode of energy. Heterotrophs need they need organic compounds. They'll need something other than carbon dioxide. So photoheterotrophs can use light as their energy source, but they need some other organic compound. We can think of this somewhat in our terms as they need to eat something. They need to ingest something as their carbon source. And then chemoheterotrophs, organic compounds for their energy source and organic compounds for their carbon source. Examples of that are animals like us. Again, we're only talking about bacteria and archaea, but as an examples, protists, fungi, some plants that ingest their organic compounds and also as an energy source and also as their carbon source. So different bacterial modes. You have this list here. You also have a great list in your book. So just review it a couple of times. Auto is without consuming something outside of itself. And photo, you can put that with light, and chemo with chemicals. That will help you at least remember the top two. In terms of oxygen, not all bacteria use oxygen, some do. We divide the bacteria and archaea into those, or the prokaryotic species, into those that use oxygen, those that don't use oxygen, and then of course there's one in the middle that can either use or not use. Adaptability tends to be the key phrase all the way through this lecture. Those that use oxygen are called obligate aerobes. So they have to have oxygen in order to function. They're obligated to have oxygen in order to function. You also have obligate anaerobes. So they absolutely want no oxygen. Oxygen would actually poison them. These are the bacteria that cause fermentation. Fermentation, you can think of wine, beer, kimchi, sauerkraut, things that are fermented. Also yogurt. And then you have the facultative anaerobes that will use oxygen when it's there, or fermentation. So we'll use a combination of both. And then on the right, some prokaryotes can metabolize nitrogen. Nitrogen in a variety of ways. Particularly some things, for example, one of the reasons why farmers will plant certain things in their fields is because they fix nitrogen, that fix nitrogen into the soil. They'll convert atmospheric nitrogen into ammonia, and in particularly in terms of bacteria in the soil, that will then put that nutrient nitrogen into the soil for the next set of plants that are put in there. Other types of metabolic cooperation that bacteria have are making these colonies called biofilms. So you have, it's a surface, it's usually a sticky surface that will allow this bacteria to form a colony until the colony becomes too big to support itself, basically. This is for growth, and it's also for protection. We're going to end here and pick up on this one on the next one.